Yeah, yeah. Yo, John.
Hello, any questions?
Nope.
I've got some bad news, and that is the lecture Zoom file did not make a video, meaning it failed to make a video. This is the first time it's failed in the lecture. Um, so Zoom is really not working well, and I will contact them and see what to do. But at the moment, I'm having trouble making the movies from our Zoom sessions. Okay, any question about any of that? For you guys, you're seeing it live, so it's not as big of a deal for you guys. But for the rest of the class, this is a real big problem. And I've never had Zoom give me so much trouble. Any questions about anything before we begin? All right, if not, let me share my screen and we will begin. So today is July 27th, and we're supposed to be talking about Lab 5. Let me remind you, you need to read about your unknown project instruction files. You can find those files on Canvas. Let me see if I still have that. I think I do. I think it's under week 4 lab. Maybe it's week 5 lab. Yeah, week 5 lab. The unknown instructions. Any question about any of that? If not, let's begin the lab. Is it lab 5? Is that right? Yeah, lab 5. There's the worksheet. Remember, when you turn in your lab, to always turn in the worksheet. Do not turn in the lab module. If you turn in the lab modules after I've warned you one time, I will apply a penalty. Because if everyone turns in the lab module, my Canvas website would soon become full, and then you guys would not be able to turn in your assignments. I think this is it. Nope, that's not it. There it is. All right, there's a little bit of reading to do, but not much. Read about culture media and read about obtaining and pure cultures. Both of them are really, that's only a page and that's a few pages. But lab 5 is the cultivation of bacteria, different culture media, inoculation, and isolation. The learning objectives for this lab is first, understand the importance of proper specimen collection and transport to the lab. 2, be able to explain the 5 principal steps used by microbiologists to process specimens. 3, differentiate between the various types of culture media. 4, be able to explain the purpose of streak plate culture technique, aka streaking for colony isolation. Does AKA, is that an American abbreviation? Does anybody know, or is that Australian? I'll have to look that up someday. 5, be able to define the following terms: what's selective media, differential media, and reducing media. Any question about our objectives? So, in introduction, the ability to isolate pathogens from a clinical specimen and one that's submitted for culture is only as good as the collection technique and the timely transport of the specimen to the lab. Some microbes are sensitive to desiccation, meaning they dry out, and some are sensitive to exposure to oxygen, like anaerobes. And some require a special transport medium to keep the organisms alive and then grow them. Once a specimen is received in a lab for microbiological culture, microbiologists employ the 5 principal I steps to process it. First, there's inoculation. 2, there's isolation. 3, there's incubation. 4, there's inspection. And 5, there's identification. So inoculation is just placing the specimen on an artificial culture medium to initiate its growth. Isolation is to spatially separate out the microbial growth into distinct colonies. Incubation is placing the culture in an environment having conditions to facilitate its growth. We usually incubate our microbiological cultures in an incubator. And we'll talk a little bit more about those. 4, inspection. Then you examine the culture first for the presence of something growing, and then the presence of microbes. And then 5, you identify the microbes. You have to usually perform specific tests to ID any pathogenic microbe that has been isolated. Meaning you just can't look at it and say, yep, that's a pathogen. You have to run some tests. The 5 steps will be described in detail after discussing different types of culture media. So we have different types of media that we can grow our cultures in. You're already familiar with general purpose media, which allows many things to grow, but not everything, like not anything that's finicky or fastidious. And some general purpose media include NA, is NAYE, nutrient agar with yeast extract, and then TSA, or tryptic soy agar. These media are cheap, they can grow a lot of different things, and they are non-selective. Any question about any of that? To promote fastidious microbes, these are organisms that require specific nutritional additives. You have to use an enriched general purpose media, such as blood agar, which has 5% sheep's blood incorporated into, I think it's tryptic soy agar, to help the organisms to grow. And we usually call those blood agar plates, or BAP for short. And another enriched general purpose media is chocolate agar plate, or CAP for short. Chocolate agar plates and blood agar plates are actually the same, meaning you have the media and you add blood to it. The only difference is that the blood agar plates, the agar is sterilized in the autoclave, allowed to cool, and then the blood is added while the agar is cool, but still liquid. And then the agar will then harden around the blood, and you have intact red blood cells in the agar. The chocolate agar plates, the blood is added immediately after you pull the agar out of the autoclave. And so this is way too hot for the blood cells, and they will lyse, spilling their nutrients throughout the plate. And it gives the culture a different color that's brown. And somebody thought that looked like chocolate, and so they called it chocolate agar. There is no chocolate in the chocolate agar plates. There's only blood, and the blood was added when it's hot, and so it lysed, and it makes the color look brown instead of red, which blood agar plates do look red. All right, so those are two different types of media. Let's talk about selective media. A selective media is one that contains a chemical that inhibits or halts the growth of some organisms, but does not inhibit other organisms. You should keep in mind two things when using selective media. First, the selection may not be perfect, meaning given enough time, organisms may grow and form visible colonies when they are being selected against. So selection is not necessarily perfect. And second, even organisms that are supposed to grow on the selective media may have slightly smaller colonies where the colonies are growing a little slower than they would on a general purpose media. Any questions about selective media? An example of a selective media is a 7% sodium chloride. Most bacteria cannot grow in 7% sodium chloride. So this will select for organisms that tolerate fairly high salt, 7% sodium chloride. Something like E. coli will not grow, but something like Staphylococcus aureus will. Any question about any of that? Another type of media we have is a differential media that contains chemicals that produce visible differences among different groups of organisms growing on the culture. These differences are unrelated to how well the organisms grow in the medium. But when you look at them, they can be visually distinguished from each other, meaning you have two different cells, you grow it on a differential media, and the two cells will look different or grow differently on the plates. It could be a difference in the pH. It could be a difference in just the way the media looks. One media, which is differential, is blood agar. And here you can see three different growths showing three different characteristics. And we'll talk about that in a little bit. The point is blood agar can be differential. It is also an enriched media because it supplies nutrients for fastidious organisms to grow, like Streptococcus pyogenes cannot grow on a general purpose media plate because it requires additives, which are not present. But you can get it to grow on a blood agar plate because Streptococcus pyogenes will lyse the red blood cells and release the nutrients so that the Streptococcus pyogenes can use those nutrients and then grow. Any question about any of that? All right, another media we're going to talk about is a reducing media. And this media promotes the growth of anaerobic bacteria because anaerobes require low or no oxygen in order to grow. And a thioglycolate media would be an example of a reducing media. It's important to note that a culture medium can be classified in more than one category. I already told you blood agar is both an enriched media as well as a differential media. Another one is MacConkey's agar, is both selective and differential. Here we're looking at MacConkey's agar, and MacConkey's agar is selective for gram negatives. It contains bile salts and crystal violet in it. That's why it has a purple color. And that prevents the growth of gram positives. Gram negatives can grow. And so if the organism can grow on MacConkey's, it's most likely a gram negative. There are always a few exceptions, like there are just a few gram positives that can grow on MacConkey's. Not very many, but there are a few. And then there are some gram negatives that cannot grow on MacConkey's. But most gram negatives can grow on MacConkey's, and it's selective so that only gram negatives can grow. It's also differential in its ability to look at lactose fermentation. If the organism can grow on the plate and it does not ferment the sugar lactose, the organism will be its normal color, which in this case is white or off-white. If, on the other hand, the organism ferments the sugar lactose, it will make the organism and the agar around the organism look pink. And it's because the sugar fermentation produces an acid and the pH becomes pink when it is acidic. Any question about any of that? So MacConkey's is both selective and differential. Blood agar is both enriched media, meaning it can allow something to grow that would not grow on a general purpose media. It also is differential in that it can distinguish between different hemolytic activity of the bacteria. Let me blow this up a little bit. Scroll over here. So this one is showing the first hemolytic activity. Hopefully that's going to go out of there. Oh, there it goes. The bacteria is white, the blood agar is red, the blood and the redness continue all the way up to the cells. This is gamma hemolysis, which is also called no hemolysis. On this one, the bacteria is off-white in color, and the blood is red. And you'll notice as we're coming up to the bacteria, we have a darkening, which is in a thin ring around the cells. The cells are off-white. The dark area is partially lysed blood, and then the unlysed blood is red. So inside the alpha, it's dark, and that's because all of this region is showing alpha hemolysis, which is the same as partial hemolysis. Whenever you lyse some of the blood, but not all of the blood, you either see the blood agar plate becoming darker, or it may appear greenish in this area. Any question about any of that? All right, the third example is shown here, and the bacteria is white on the B or beta here. And then this clear region around the B or beta is the complete hemolysis of blood, and that we call beta hemolysis, the complete lysing of blood. If you were to hold this plate up to the light, you could actually see the light bulb in this region of the plate. You wouldn't see it here or where the bacteria is here, but you could see it here. Any questions about blood agar plates and the difference between the differential hemolytic activity as well as blood agar is an enriched media plate? No questions. Let me make one other comment, and that is whenever you have beta hemolysis, you'll note that there's a ring of alpha hemolysis further out from the beta hemolysis. And I've always seen that. It's because this region, there's something being secreted by these cells, which is lysing the blood. But in this region, it's so little that only some of the blood is lysed. But there is a alpha hemolysis ring around the beta hemolysis. All right, this table is summarizing the different types of media, the general purpose media, tryptic soy agar, nutrient agar, and then nutrient agar with yeast extract. It's also got the enriched media, which is blood agar and chocolate agar, and it's enriched because it has blood added to the general purpose media. Blood agar is also differential in that you can see different hemolytic activity of different species of cells, meaning there's beta hemolysis, alpha hemolysis, and gamma hemolysis. And then there's McConkey's agar or MAC, EMB plates or eosine methylene blue, or HE plates, that's hectone agar. All of these are selective and differential. They are all selective for gram-negative bacilli to grow, and they're differential on their basis of lactose fermentation. McConkey's, if it ferments the sugar lactose, it'll be pink in color. EMB plates, if it ferments the sugar lactose, it'll either be dark, like black, or a metallic green color. And then hectone enteric agar is differential. If it ferments the sugar lactose, it'll be salmon colored. If you don't know what salmon is, it's an orangish pink color. And then we have selective media. We only have one that we're going to talk about. That's PEA, or phenyl ethyl alcohol. The phenyl ethyl alcohol select against gram negatives. So if the bacteria grows on PEA, it's likely to be gram positive. And then there's mannitol salt agar, which is both selective and differential. It's selective in that only organisms that can grow at fairly high salt, I think it's 7.5% salt. And then it's differential on the ability of the bacteria to ferment the sugar mannitol. If it can grow on the plate, it means it tolerates the salt present in mannitol salt agar. And if it's differential, I mean, if it does grow, then you can differentiate between those that ferment this ferment the sugar mannitol and those that do not ferment the sugar mannitol. If it does ferment the sugar mannitol, the bacteria will be yellow or will have a yellow halo around it, meaning the agar will turn yellow around the colony. If it does not ferment the sugar lactose, that won't happen. It won't be yellow. Any question about any of that? All right. So once again, we're going to look at inoculation, isolation, incubation, inspection, and identification. Inoculation is just inoculating the plate with the culture. Isolation is isolating the colonies, and you need to do that 2 times to get a pure culture. Louis Pasteur came up with the concept of working with pure cultures, and he came up with a procedure for obtaining them. It was a very difficult procedure. You took your bacteria in a liquid culture, and then you made several different dilutions of that bacteria. and then you took each of those dilutions and you plated them 10 times into a test tube. If one of the dilutions had 1 tube that had growth and 9 tubes that had no growth, Louis Pasteur correctly concluded that the 1 tube with growth would likely be a pure culture. Doesn't mean it's always a pure culture, but most of the time it is a pure culture. It was very labor intensive because you had to make a number of different dilutions, something like 5 or 6 different dilutions, and then you had to plate 10 tubes for each dilution. And then you had to allow them to grow and then look for one that only had 1 tube in 10 with growth. And if you had 2 tubes with growth, you were out of luck, then you had to do the experiment again. Robert Koch developed a much easier way to obtain a pure culture, and that is he streaked on a solid media, like a solid agar plate, he streaked for getting isolated colonies. And if you do that 2 times, you will have an isolated pure culture in over 95% of the cases. If you only streak 1 time for isolated colonies, only 70% of the colonies are pure, and 30% were started by 2 or more cells, and 70% is not high enough to work with in microbiology. But if you streak 1 isolated colony out on a second plate to obtain isolated colonies, and now if you take from an isolated colony, the cells from that isolated colony will be pure in over 95% of the cases. Any question about any of that? Come on. When you're streaking out for colony isolation, what you're doing is taking your loop, sterilizing it, picking up some bacteria, and then streaking in 1 quadrant. When you very first start, the bacteria will be very high, and then as you streak, the bacteria numbers will decrease, but they'll only decrease by a little bit, at most a factor of 2. Something like a factor of 2. You then take your loop and you sterilize it. I'll use the mouse here. You then take your loop and you sterilize it. And after doing culture 1, you then do sector 2. When you're doing this, this is not done quite correctly, and that is you should streak out culture 1. It'll be a little higher right here than it will be right here. And then to streak out culture 2, you sterilize your loop, killing all the cells that are on it, and then come into start quadrant 2. And then where you get your cells is you move into quadrant 1, and you only go where the cells are at low number, like right here, and then move back into quadrant 2. And it's true, you do move into quadrant 1 about 3 times. Don't do it more than 4 times, but you should do it more than once. You should do it about 3 times or 4 times. And when you come into quadrant 1, you should do it where the cells are a little dilute, meaning the end. Don't do it in the high areas. So this shouldn't come all the way up here. It should not be any further than there, and preferentially it should be only to here. Okay? But you pick up your cells for quadrant 2 from quadrant 1, and then streak out quadrant 1. You then take your loop, and then you're sterilizing it, killing all the cells from quadrant 2 that were on it. And then you start streaking in quadrant 3, which is shown correctly here. And then you come and pick them up from quadrant 2. Once again, you should not come into where the cells are high in number, this area here. You should only pick them up where they're low in numbers, this area here. And you should only do it 3 or 4 times. 1, 2, 3, 4, 5, 6. You shouldn't do it 6 times. You should only come into quadrant 2 3 or 4 times. And do it where it's lower. It's right there. Actually, for quadrant 4, this is actually shown correctly. You take your loop, then you then sterilize it. You put the loop in quadrant 4, and to get your cells, you pick them up by going into quadrant 3, where the cells are more dilute, this region here. You do not pick them up from this region here. And then you streak out for quadrant 4. The goal is that someplace along where you're streaking, you'll be putting down cells where the cells will be put down few and far apart so that you will get isolated colonies. In this case, we're getting the isolated colonies in quadrant 4, but it doesn't matter where the isolated colonies occur. If the culture were more dilute than this, you get the isolated colonies from quadrant 3. And if it's more dilute than that, I guess that's quadrant 3 here. If it's more dilute than that, you get it in quadrant 2. And if the cells are very dilute to start with, you'll get the isolated colonies in quadrant 1. Any question about any of that? So theoretically, some place along where you're doing the streaking, individual bacterial cells will be put, allowing the colonies to grow up and be isolated. So as we were streaking, 1 or 2 individual cells, or maybe a clump of cells, went there, and then 1 or 2 or a clump of cells went there, and then they grew up and formed isolated colonies. Now, to make sure you have a pure culture, what you do is you grab the cells from one colony, like right there, and then streak it out again on a second plate to get isolated colonies. And then you get an isolated colony here. You then pick from that one isolated colony where the cells have gone through 2 colony forming events, one on this plate and one on this plate. And then the chance that you'll have a pure culture is higher than 95%. And that's good enough to use in microbiology. All right. Incubation is growing the culture in an environment suitable for the culture to grow. For our incubators, they're pretty simple. All they do is they heat the culture to either 30, 35, or 37 degrees, depending on the incubator. And then we give them some water so that the growth will not dry out as quickly, or the plate will not dry out as quickly as otherwise, because we're providing some humidity. And that pan of water right there is providing the humidity. The water will evaporate from the heat of the incubator, providing the humidity. If you don't apply water, then the plates will dry out sooner. Fancy incubators like this one here can supply an oxygen concentration and a CO2 concentration. For anaerobes, for example, they have to grow without oxygen. And so you grow them in an environment where the oxygen is totally removed from the incubator, or in an anaerobic jar where the oxygen is totally removed. The problem with an incubator is you have to open the door to put things in and out, and obviously oxygen can get in that situation. So that's the advantage of an anaerobic jar, and that is oxygen will not get in until you literally open the jar up. You then inspect the cultures for visible colonies. You can look at the different colony morphological appearances to distinguish certain species of bacteria. And you can describe the appearance of the colonies by their size, their margin, their shape, their elevation. We'll talk a little bit more about the colony characteristics in just a little bit, but generally that will not be enough to identify the species. To identify the species, you will have to run specific tests to determine what species the organism is. And that's identification. So know the terms of this lab. You don't need to go through the references. And then let's talk about the laboratory exercise. I think I'm going to switch to the exercises now. Nope, I can't do that. Not yet. So we have a little video for you to watch on inoculating an agar plate using the streak plate culture technique. You only need to watch this video for 4 minutes and 15 seconds into the video. After that, it doesn't need to be watched, but you're free to watch it. You should note when they're making this plate, they're only streaking in 3 sectors, and I showed you a picture streaking in 4 sectors. It doesn't really matter how many sectors you have on a plate. Generally, people streak out 3 or 4 sectors on a plate. We then have a little video on discussing beta hemolysis, another one on alpha hemolysis, and a third video on gamma hemolysis. Go ahead and look at those. You'll need to know that for the lab. And then we have a video on MacConkey's agar. You only need to watch this one for 3 minutes and 50 seconds into the video. And then we have a little video on mannitol salt agar. We no longer have a virtual lab, which would help you actually do a virtual streaking, where you would streak out a plate on the computer. Unfortunately, the lab uses Adobe Flash, and the Flash player is no longer working. If anyone still has it, it's no longer working. And then work on the questions. Please understand you're not going to be doing any of the lab. We only have videos for you to watch, and then the lab to do, because this class is totally online. But answer the questions, describe a natural circumstances where staphylococcal species of bacteria might benefit from being salt tolerant. So come up with a natural environment. So if you say growing it in mannitol salt agar, that would not be a natural environment. So pick a natural environment where Staphylococcus will tolerate a high salt concentration. And 2, how might hemolysis benefit a hemolytic organism? So we have a bacteria, and it lyses the red blood cells on blood agar because of its hemolytic activity. Why might that benefit the bacteria? Oops, too far. Suppose we wanted to describe a possible species that could be many different species. And we have 3 different plates. Describe a scenario where you use mannitol salt agar, blood agar, and EMB plates to identify the unknown bacterial species. That is, how would you use these plates to help you identify the species? You do need to discuss 2 properties to each plate that you can use to help identify the species. Any question about any of that? If you do not have 2 properties to each plate, then you'll get a partial wrong answer. And because this question is longer and requires more writing in detail than most of the other questions, I've upped its value. Instead of being worth 2 points, this one will be worth 6 points, meaning each plate will be worth 2 points. Any question about any of that? And normally each question is worth 2 points. What is the main goal of streaking for isolation? Meaning when we're streaking out cells on a plate and we're streaking out for colony isolation, what's the main goal? And 5, what happens if you forget to sterilize the loop in between sectors? And this should be changed. Let me see if I changed it on your example. Hopefully I did, but this is the summer and I may have changed it after the summer. Can't see it, but there it is. There, I do have it in there. You need to be specific discussing each sector. So if you simply say, what happens on the plate if you forget to sterilize the loop between each sector? I can't read all of that. Scroll it over like there. And you simply say, well, you won't get isolated colonies. I'm going to tell you that's not correct. You need to discuss each sector, meaning tell me what growth you see in each sector to answer this question. Any question about that? And you have it for number 6 too. So I need to change it, my copy, but oh, I've got it changed here. 6, what happens to the growth on the plate if you transfer from the stock culture to inoculate each of the sectors? Once again, you must be specific and discuss each sector. If you just simply say you won't get any isolated colonies, that's not correct. What does each sector look like on the plate? All right. 7, suppose you successfully streak a plate and obtain isolated colonies.
Can't hear you, dog.
Was that because of the internet or what?
Yeah, it was the internet.
Okay, yeah, it just told me now the internet connection's unstable.
You're good now.
Okay. 7, suppose you successfully streak a plate and obtain isolated colonies of 2 different bacterial species. That is, from a mixed culture. How can you use this plate to create a new pure culture of only one of the bacteria? Be specific in your answer. 8, in the table below are a variety of agar plate media available to microbiologists to cultivate bacteria. Denote whether each medium is non-selective, like a general growth medium, is selective, or differential by placing an X in the appropriate column. You may use the table of media in the lab module to answer these questions. So nutrient agar, if you think it is selective, put an X there. Tryptic soy agar, if you think it's selective and differential, put an X here and here. You know, obviously I gave you wrong answers, but my point is put the X where you think they are. And then answer that. And then 9, we have 3 different plates here, and it asks you which agar plate was inoculated using a streak plate technique. Just tell me plate A, B, or C. You just need to know put the initial. And then that's it for the lab. It's 7:47. I will be here until 8:30 to answer any lab questions, but I will log off once the last student logs off. So if you have a question, make sure you log in before the last student logs off. And let me state that the lecture file in Zoom did not finalize, and so it did not make a video. And I'm not sure what I will do. I'll try and find maybe an older video and post it. I'm going to have to contact Zoom and Clark College to find out what's going on and why Zoom is not working. Any questions about any of that? If not, start working on the lab, and I'll be here until 8:30. Oh, I'm still recording. Let's stop